Support for the Journal of Clinical Oncology podcast is provided in part by AstraZeneca, dedicated to advancing options and providing hope for people living with cancer. More information at AstraZeneca-US.com.
Hello and welcome to JCO Precision Oncology Conversations, where we bring you engaging conversations of clinically relevant and highly significant JCO articles. I'm your host, Dr. Rafeh Naqash, social media editor for JCO Precision Oncology and assistant professor at the Stephenson Cancer Center at the University of Oklahoma. Today, we are excited to be joined by Dr. Jun Gong, associate professor, Division of Medical Oncology at the Cedars-Sinai Medical Center, and lead author of the JCO Precision Oncology article entitled Phase 2 Study of Erdafitinib in Patients with Tumors Harboring FGFR Amplifications. Results from the NCI-MATCH ECOG ACRIN trial EAY131 subprotocol K1. And Phase 2 Study of Erdafitinib in Patients with Tumors with FGFR mutations or fusions. Results from the NCI-MATCH ECOG ACRIN trial EAY131 subprotocol K2. Our guest disclosures will be linked in the transcript. Dr. Gong, welcome to our podcast and thank you for joining us.
Thank you, Dr. Naqash and JCO Precision Oncology for having me.
We are excited to be discussing some interesting aspects that you have led and published on from the NCI-MATCH trial. And we were trying to understand from a background perspective, since this master protocol has been going on for quite some time, could you give us a little bit of a background for the sake of our listeners, what the NCI-MATCH is and what were the specific objectives for these two sub-protocols?
Yes, of course, Dr. Naqash. So as you may all know, the importance of targeted therapies in the current era of precision oncology. And on that backdrop, the NCI-MATCH was a national multicenter study designed essentially to look for signals of efficacy across various solid tumor and hematologic malignancy types with the focus on specific mutations. The master protocol is unique in that there are several arms to the trial, each targeting a specific potential targetable alteration using available agents that are available in cancer today.
Excellent. Thank you for that background. And I know this master protocol has been going on for quite some time with different sub-protocols. I believe some of them are immunotherapy based also. Now, you've led two important sub-protocols, which is the FGFR amplification and the FGFR mutation or fusion. And there are some differences from what I gather on responses for the fusions versus the amplifications or mutations versus the amplifications. Could you first delve into the first paper of the fusions, describe what were the tumor types? As you mentioned, the paper, some tumors were excluded. What was the reason for exclusion of some of those tumor types? Why did you want to study the fusions and mutations versus the amplifications separately? What was the background for that? Could you highlight some of those points for us?
So firstly, as a kind of a more background, FGFR has been a recognizable target for a couple of tumor types. And if you look at the kind of the broad landscape of FGFR alterations, they occur in about 5 to 10% of the cancers, majority being FGFR amplifications, actually, and mutations and rearrangements following second and third, respectively, and most commonly identified alterations. With that being said, FGFR mutations and rearrangements have actually already been established in a couple of tumor types. Actually, the first FDA approval for an oral FGFR inhibitor was Erdafitinib, which was the agent used in both of these back-to-back trials. However, Erdafitinib was firstly approved in urothelial carcinoma. And then since then, there's been kind of an explosion in oral FGFR inhibitors targeting fusions and mutations in other cancer types, such as cholangiocarcinoma. And more recently, there was even an FDA approval in a myeloid malignancy as well. So we used Erdafitinib, being that it was kind of the first FDA-approved orally available agent to target this alteration. And we conducted the two back-to-back studies in recognition that although rearrangements and mutations have already been established in certain tumor types, we were more interested as well in looking at the more common FGFR alteration, that being amplifications. However, the efficacy in that was a little unknown. And so these two separate subprotocols developed. K2, which was to look at FGFR mutations and fusions in tumor types excluding urothelial carcinoma to look if there is a signal of efficacy beyond currently FDA-approved indications, and amplifications as a separate cohort.
That's a very good explanation of why you concentrated on the tumor types in these protocols. Now, going back to subprotocol K1, could you tell us on what were some of the tumor types that you did include, and what was the sample size, and what was the hypothesis for the sample size as a meaningful level of activity that you wanted to see and would have potentially led to a bigger, broader trial?
So K1, subprotocol K1, was the arm investigating Erdafitinib in those with FGFR amplifications. And these were predefined on the NCI-MATCH protocol, looking at FGFR 1, 2, 3, and 4 amplifications, essentially. These were allowed to have local testing through a local CLIA certified assay, but then they needed to be confirmed on a central assay, which is the NCI-MATCH OncoMine assay. The statistics are kind of uniform for the NCI-MATCH trials, and the goal was at least 31 patients, with the hypothesis that if the response rate was 16% or more, this was considered a signal of activity. However, there was an additional protocol-specific requirement in that if the sample size was fewer than 31 patients, then the primary efficacy population would be assessed against a null hypothesis overall response rate of 5%, meaning that if it was less than 31 subjects, an overall response rate of greater than 5% would be defined as positive. And again, the NCI-MATCH was uniform. Secondary objectives included progression-free survival, overall survival, and safety and toxicity. With that being said, K1, we originally began accrual. The NCI-MATCH actually launched in 2015, but in the subprotocol K1, 35 patients were initially enrolled in the study. If you go down kind of the eligibility criteria, however, a lot of these patients did drop out due to lack of central tumor confirmation and various reasons. Ultimately, 18 patients were included in the pre-specified primary efficacy cohort.
Thank you. I did see for subprotocol K1, you mostly had stable disease in a couple of patients, no responses. And I think one individual with breast cancer had a prolonged stable disease. Now, from an FGFR amplification standpoint, did you or were you able to correlate? Now, again, this is not objective responses. It's not a partial response or a complete response. Was there any correlation from the level of amplification, the duration of stable disease?
That's actually the core of our discussion of why K1, despite a kind of a variety of basket of solid tumor types, some where preclinical data had suggested FGFR amplifications could be targeted, that K1 ultimately was a negative trial with a 0% response rate. And we dive in that although we included as an eligibility criteria a copy number variation of 7 as the threshold for amplification, we realize if you look at some of the literature out there, that even in the FGFR 1 and 2 amplification cohorts, where these are kind of the more common cohorts of amplified tumor types that have been targeted, that you really needed a high level amplification, more than 99% of tumor cells to be amplified in the previous studies to actually generate a response. And the thought was that we assumed that FGFR amplification would lead to protein expression and dependence on FGFR signaling, providing sensitivity to FGFR inhibition. But I realized that there is a certain degree where high level amplifications need to happen, and it may not be also for all FGFR amplifications. We looked into the literature that FGFR 1 and 2 were the more commonly studied FGFR amplifications. FGFR 1, if you actually look at the amplicon structure, it tends to amplify a lot of other genes because it's such a huge amplicon structure. But FGFR 2, very shorter and centered on a really just FGFR 2 with few other genes co-amplified. So actually in the literature, they've been already saying that maybe FGFR2 amplification tumors are more readily targetable based on the robustness of evidence rather than FGFR1. But across all of these, the higher the level of amplification, seems the more targetable.
Those are interesting discussions around protein expression on the tumor that could imply therapeutic vulnerability. So I've always thought about it, whether trials like NCI-MATCH or TAPER, for example, ASCO TAPER, would be perhaps more informative if, on a secondary analysis standpoint, proteomics is something that could be done on the tumor tissue. Because similar to NCI-MATCH, ASCO TAPER has other subprotocols where some of these mutations or amplifications don't necessarily result in anti-tumor responses. But I think from a biology standpoint, as you mentioned, a certain amplification might correspond to RNA expression, and that might correspond to protein expression, which is downstream. So looking at that would be something interesting. Have you planned for something like that on these tumor specimens if you have biobanked any of those specimens?
I think that's a great future direction. And I know you, Dr. Naqash, being involved in so many cooperative group trials, I think it is possible, but it really depends on good trial planning from the onset. When designing such massive trials like this, I think the more important thing is if your trials are negative, but they are informative for the field, to go back and have these post-hoc available biobanks that you said. And I think having it integrated firstly is way more efficient than to have kind of an amendment kind of going through halfway or when the trial is started. That could be a little bit more logistically difficult.
I completely agree. And you mentioned cooperative groups. I think we've been discussing, and I'm pretty sure you have also, there's a lot to be learned from clinical trials that are negative. We often in the academic or non-academic setting end up not publishing some of those negative results, pharma or cooperative group-based studies. And I think the resource is the specimens, and the negative results could correlate to some other novel findings if some of those exploratory analyses are done in the appropriate manner. Now, going to the drug itself, Erdafitinib here, it's a FGFR, pan-FGFR inhibitor. Is that something that you think is a limitation for in the drug development space? I do early phase trials, and I'm pretty sure you do a lot of these basket early phase trials. Is that something that you feel is a limitation when you have a drug that targets different mutations or different protein changes of the same gene or different amplifications? Could that be a reason why something like this doesn't necessarily work, because it doesn't have as much of specificity against that isoform as one might need to inhibit the downstream kinase activation?
That is also a great point. The NCI-MATCH subprotocol K1 and K2 used Erdafitinib, which was the first FDA-approved FGFR inhibitor. But as many of the listeners and yourself may know, there have been newer iterations and next-generation development of the FGFR inhibitors. And it's very fascinating, the tyrosine kinase inhibitors. With each iteration, you seem to have a little bit more potency and the ability to bypass some of the resistance mutations, almost paralleling kind of the lung cancer space where we kind of follow that, and they've been kind of the pioneers in that space. And to your point, yes, we considered, the NCI-MATCH was developed nearly a decade ago, and the available agents at that time. Would it have changed the findings if we used a kind of a newer generation or more potent FGFR inhibitor? It's possible. I think especially in the K1 cohort with the amplifications, we even suggested in the discussion of the paper, future directions is that one way to kind of bypass the amplification issue is to use more potent and specific FGFR inhibitors. And so I think that's very possible that you highlight this point.
And for the sake of our listeners, June, especially trainees, could you highlight what are currently some of the FDA-approved FGFR inhibitors and what tumor types are they currently approved in?
The first one, as we've hinted, was in treatment of refractory, essentially, urothelial carcinoma with FGFR mutations and rearrangements, mainly 2 and 3. And this is where oral Erdafitinib was approved. And it's interesting, I kind of teach my fellows and our health staff that Erdafitinib is interesting in that its FDA label insert requires a starting dose of about 8 milligrams daily, and it's a 28-day cycle. But during the first 14 days, we're really looking at the serum phosphate levels. If they are within a certain level, if they are within 5.5 to 7, for example, you continue the current dose. But if they are less than 5.5, the FDA label actually mandates that you increase it to 9 milligrams oral daily continuously. And this is biologically logical to me. FGFR is located to the renal tubules. And so this is a major phosphate kind of metabolism pathway here. And so you're using that as a surrogate, essentially, of its, if the right dosing is achieved. And so that's unique. And then the subsequent kind of FGFR inhibitors that came about, you had a couple in cholangiocarcinoma, where unlike urothelial carcinoma, where it's about 30% of the time you'll find the FGFR alterations of target, it's about half of that, 15% in cholangiocarcinoma. And it's mainly intrahepatic cholangiocarcinoma in that sense. And here you have pemigatinib, which is one of the FGFR inhibitors approved for cholangiocarcinoma. And then you also had infigratinib, which is approved. But however, infigratinib eventually had their FDA label pulled. It was withdrawn by the company. I think it was in 2022. And then more recently, you had even a more potent FGFR inhibitor in cholangio approved, and futibatinib. It's interesting that with these more later generations of FGFR inhibitors, they do show a correlation with phosphate levels, but they don't have that specific kind of dosing early on in the first cycle, like erdafitinib. And so it's interesting to see that with the later generations, you're seeing more potency as well.
Thank you for that overview, which I'm sure most of the trainees appreciate, since this is an up-and-coming field in the space of precision medicine, especially FGFR. From a side effect profile standpoint, you mentioned phosphate issues. Do you think that is a drug class effect here, or is that an FGFR receptor subtype effect, depending on which FGFR receptor, 1 or 2 or 3, that is being targeted?
I do think this is a class effect that you'll see across a lot of the trials, where phosphorus elevations or decreases are going to be probably your most common treatment-related adverse event. And I actually emphasize this is probably one of the most trickier side effects of this class, where you're almost having to have to monitor the phosphorus levels pretty routinely, pretty closely. And you also have other class effects on the nails. There's some rare retinal ocular toxicities that's unique to the FGFR class as well. And so it's a very exciting class of compounds, but it does require some close monitoring of some unique class effects, as you hinted.
And based on the results from your K1 subprotocol, are FGFR inhibitors still the approach within, let's say, cholangio or urothelial with FGFR amplifications? Is that still something that has been established and seen from a clinical response standpoint?
The FDA approvals are really for mutations and fusions. So this K1 subprotocol essentially, I think, provides one answer that we've been all wondering about for the longest time. Hey, could amplifications be targeted as well? Unfortunately, we didn't include urothelial carcinomas in this study because of the FDA approval, but from a kind of a basket solid tumor perspective, I think this really dampens the enthusiasm. As of right now, it really is fusions and mutations that are targetable. Amplifications need further investigation before becoming an established, I think, in solid tumors.
Going to the discussion with the second K2 protocol, which is mutations and fusions, can you highlight again what were the tumor types there? What are some of the clinical outcomes that you saw, and any unique insights on certain mutations or protein changes that were a little more relevant than some others?
Sure. So this is the parallel study to K1, in that now we are looking at fusions and mutations of FGFR 1, 2, 3, and 4. And essentially, we again excluded those with urothelial carcinoma, given the FDA approval for erdafitinib in this trial. The trial actually opened, and then the FDA approvals for the FGFR inhibitors for cholangiocarcinoma happened. So actually, this trial didn't really exclude those with FGFR mutated or rearranged cholangiocarcinoma as well. If you look at kind of the breakdown of the cohort in K2, you saw, again, a good mix of breast cancers, or a couple of breast cancers, or a couple of gynecologic malignancies. There was a couple of head and neck cancers. There were several brain tumors as well. There was one lung cancer. There was noted 4 intrahepatic cholangiocarcinomas. Again, we could not exclude those due to the fact that the trial had opened and it was accruing when the FGFR inhibitors approved for cholangiocarcinoma happened. Similar kind of design, phase 2 single arm, open label of Erdafitinib. And again, the same statistical design was implemented in that if it's higher than 31 patients, 16% overall response rate was the primary endpoint goal. If it was less than that, it was against the 5% overall response rate. And here in K2, 35 patients were enrolled and 25 patients were ultimately included in the primary efficacy analysis. So because it was fewer than 31 in the primary efficacy cohort, it followed the NCI-MATCH pre-specified primary endpoint goal of 5% or higher. And here, in a heavily pretreated cohort of more than 50% of subjects had received prior than 3 or higher lines of therapy, the overall response rate essentially confirmed was 16% with the p-value of 0.034, which met the positivity cutoff of 5%. However, an additional 7 patients experienced stable disease as best confirmed response. And it's important to note that 4 of these were grade 4 glioblastomas who had prolonged progression-free survival. And so ultimately, this trial was positive in meeting the endpoint that outside of urothelial carcinoma, could FGFR inhibition be pursued in other tumor types that had FGFR rearrangements or fusions?
You mentioned glioblastoma, which is an area of huge unmet need. Do you think a trial like this as an upfront approach in glioblastoma, perhaps maybe after Temodar, could be a more meaningful way of using the strongest, more precise therapy earlier on when there are certain mechanisms that inhibition of which would result in anti-tumor responses? Do you think doing this earlier on rather than second, third, fourth line would be perhaps more intriguing in some ways?
I think you've hit upon several key points there. Firstly, just the high unmet need in glioblastomas in general. And then again, to us, although it was a stable disease, it was quite noticeable that 4 of these occurred in IDH1 and 2 wild type brain tumors. We kind of discussed that in the discussion as well. And of these, we actually realized that in the preclinical and other published literature space, that for some reason, IDH1 and 2 wild type gliomas tended to have more FGFR alterations, while 0% were found in IDH1 and 2 mutant high-grade gliomas. So I think there is something hypothesis generating to come out of this study as well, even though there were stable disease, and that you may be we may be able to have future studies to select for a specific niche of glioblastomas. And as to your point, Dr. Naqash, I think if you can have a design trial looking for the specific molecular subsets, I think it's wide open for trials of this nature in the first line, second line, or refractory space. Even piggybacking to cholangiocarcinoma and UC, they're now looking at these in the neoadjuvant and adjuvant space as well. So I think you can identify the subset. It's wide open out there.
I completely agree. I remember my program director a few years back when immunotherapy was in the metastatic setting was very exciting. And he gave a talk where he said, early, earlier, earliest. And the more early, the better it seems. So I'm guessing that it's probably something similar for precision medicine-based approaches, like targeting FGFR perhaps earlier. So what is next for some of these 2 studies or these ideas that have come out of these 2 studies? Are you trying to develop something subsequently, or is the NCI-MATCH looking at it from a certain perspective? Or what would you want to do as a next step, ideally, if you had the funding and the pharma support?
That's the $1 million question. So from just the broad strokes, I think what these 2 back-to-back papers and studies comment is that amplifications may not be the more targetable of FGFR subset, but there is avenue for improvement there and further investigation. FGFR fusions and mutations, however, seem to go along with what we know in some of the FDA-approved types now. Now, the next step is in the era of precision oncology is, could we expand the labeled indications now to other subtypes with FGFR fusions and mutations. And this is, I think, following precedent. You and the audience may know that there's a lot of different tumor agnostic approvals now for both immunotherapy and other targeted therapy types. So I think the goal of this study was to provide momentum for perhaps advancement into a tumor agnostic indication for FGFR inhibitors. And we do cite in the K2 manuscript the results of a phase 2 study that was also published around the time we were writing this study up. It was the phase 2 Ragnar study, and that enrolled patients again with FGFR fusions and mutations. And that trial was positive too. That one was a larger study of 217 subjects. We highlight some differences in study populations as to why maybe the difference in responders were detected. Both were positive studies. It was reassuring that the overall survival in both studies were about the same. And again, I think they don't compete. I rather think they complement each other in kind of just providing this body of evidence that maybe at one point the FDA should be approached with this evidence for a tumor agnostic indication so that more patients with this subset could be benefiting.
Well, excellent. Thank you so much, Jun. Now, could you tell us briefly what your background is, where you train, and your interests, and how you balance clinical research with some of your personal interests?
Sure. Thank you for that interest. I did my training in medical school in New York. I went to New York Medical College, and then I did my residency at Cedars-Sinai for medicine, actually. And I went to City of Hope for fellowship, where I was trained in GU by Dr. Monty Powell, who many of us are familiar with. And my GI training was with Dr. Fakih at City of Hope. And since then, I returned back to Cedars-Sinai, where I kind of serve as a dual GI/GU focused medical oncologist. I do clinical trials in both and translational science, really focused on targeting tumor metabolism in both as well. My advice to the listeners and trainees, and I tell my own fellows this, I think it's very rare now, unless you're in phase 1, to kind of do a dual focus. So I actually emphasize to my trainees that it's the more focused you can be, the better, unless you are going into phase 1, for example. With that, you can kind of hone in, develop your craft. But then again, I have known several mentors who do multiple tumor types, but I think the more traditional mechanism is to focus as much as you can, as my advice for the listeners.
Well, thank you again, Jun, for all those interesting scientific and personal insights. We appreciate you in working with JCO Precision Oncology for both of your manuscripts. This is the first time we have ever invited lead author for 2 manuscripts at the same time. It's always good to be the first in something. And I learned a lot, and hopefully our audience would have learned a lot.
Thank you, Dr. Naqash, for having me. It was a pleasure speaking with you and the crew.
Thank you. Thank you for listening to JCO Precision Oncology Conversations. Don't forget to give us a rating or review, and be sure to subscribe so you never miss an episode. You can find all ASCO shows at asco.org/podcast.
The purpose of this podcast is to educate and to inform. This is not a substitute for professional medical care and is not intended for use in the diagnosis or treatment of individual conditions. Guests on this podcast express their own opinions, experience, and conclusions. Guest statements on the podcast do not express the opinions of ASCO. The mention of any product, service, organization, activity, or therapy should not be construed as an ASCO endorsement.